The discovery of CRISPR-Cas9 has been a revolutionary moment in science. This groundbreaking tool has opened up new frontiers in genetic research, allowing scientists to explore the very building blocks of life with unprecedented precision. It's like having a pair of tiny scissors that can precisely cut and edit the very code of life. Our DNA. Imagine being able to remove a single letter from a vast encyclopedia and replace it with the correct one, fixing errors that cause diseases. This groundbreaking technology allows us to target specific genes, correct genetic defects, and potentially cure diseases that were once considered incurable. The possibilities are endless, from treating genetic disorders to enhancing crop resilience in agriculture. For centuries, scientists have dreamed of having the ability to manipulate genes with such precision. Early geneticists laid the groundwork, but it wasn't until the advent of CRISPR-Cas9 that these dreams began to materialize into tangible results. Now, with CRISPR-Cas9, that dream has become a reality. Researchers around the world are harnessing this technology to push the boundaries of what we thought was possible in genetic science. This technology is rapidly changing the landscape of medicine, agriculture, and even our understanding of life itself. In medicine, CRISPR is being used to develop therapies for genetic diseases, while in agriculture it is helping to create crops that are more resistant to pests and environmental stresses. The implications of CRISPR are far-reaching, offering hope for treating a wide range of genetic diseases. Scientists are optimistic that this technology could one day lead to cures for conditions that have long eluded medical science. From cystic fibrosis to sickle cell anemia, CRISPR has the potential to alleviate human suffering on a global scale. Patients who once had little hope are now seeing the possibility of a future free from the burden of genetic diseases. The recent FDA approval of the first CRISPR-based therapy marks a pivotal moment in this genetic revolution. This approval not only validates the years of research and development, but also paves the way for more CRISPR therapies to enter the market, bringing new hope to patients worldwide. CRISPR-Cas9 might sound complicated, but its basic principles are surprisingly simple. This revolutionary technology has transformed the field of genetics, offering unprecedented precision in editing DNA. Imagine a pair of molecular scissors guided by a GPS system. These molecular scissors can be directed to any location within the vast expanse of the genome. That's essentially what CRISPR-Cas9 is. It's a tool that allows scientists to make precise cuts in the DNA, enabling them to modify genes with incredible accuracy. The scissors in this case are a special protein called Cas9, which has the ability to cut DNA. This protein is derived from a bacterial immune system where it originally served to defend against viruses. The GPS is a short RNA molecule called guide RNA. This guide RNA is crucial because it directs the Cas9 protein to the exact spot in the DNA that needs to be edited. This guide RNA is designed to match a specific DNA sequence in the gene we want to edit. It acts like a homing device, ensuring that the Cas9 protein cuts at the right location. When introduced into a cell, the guide RNA leads the Cas9 protein to the target DNA sequence. This process is highly efficient and can be used to target virtually any gene. Cas9 then makes a precise cut at that location. This cut is the first step in the gene editing process, creating an opportunity for scientists to make specific changes to the DNA. This cut in the DNA triggers the cell's natural repair mechanisms. The cell attempts to repair the break, and scientists can harness this process to introduce new genetic material or correct mutations. Scientists can exploit these repair mechanisms to either disrupt a faulty gene or insert a new, corrected piece of DNA. This ability to precisely alter the genetic code has vast implications for treating genetic disorders. This precise gene editing capability is what makes CRISPR-Cas9 such a powerful tool for research in medicine. It holds the promise of curing genetic diseases, improving crop resilience, and even combating climate change by altering the genes of organisms to better adapt to their environments. In a landmark decision, the FDA recently approved the first-ever CRISPR-based therapy for sickle cell disease and beta thalassemia. These inherited blood disorders affect millions worldwide, causing debilitating pain, organ damage, and reduced life expectancy. The therapy, known as exa-cel, offers a potential one-time cure for these debilitating conditions. Exa-cel works by editing the patient's own blood stem cells. These stem cells are responsible for producing red blood cells. In sickle cell disease and beta thalassemia, a defective gene causes red blood cells to become misshapen and dysfunctional. Exocell corrects this defective gene in the stem cells, allowing them to produce healthy red blood cells. This FDA approval is a watershed moment in the field of gene editing. It paves the way for a new generation of CRISPR-based therapies that could revolutionize the treatment of countless genetic diseases. Section 4. How CRISPR therapy works, a step-by-step guide. The process of CRISPR therapy, while complex, can be broken down into several key steps. First, identifying the target. This is a crucial initial phase where scientists focus on pinpointing the exact gene responsible for the disease or condition in question. Scientists first identify the specific gene that is responsible for the disease. This involves extensive research and analysis of genetic data to ensure accuracy. This gene becomes the target for CRISPR editing. By isolating the gene, researchers can focus their efforts on modifying or correcting it. Next, designing the guide RNA. This step is essential as the guide RNA will direct the CRISPR machinery to the exact location in the genome. A guide RNA molecule is designed to match the DNA sequence of the target gene. This involves creating a sequence that is complementary to the target DNA. This guide RNA will act as the GPS to direct the Cas9 protein. The Cas9 protein is an enzyme that can cut DNA at specific locations, guided by the RNA, then delivering CRISPR-Cas9. The CRISPR-Cas9 complex, which includes the Cas9 protein and guide RNA, is delivered to the patient's cells. This delivery is a critical step to ensure the CRISPR components reach their target. This can be done using various methods such as viral vectors or nanoparticles. Each method has its own advantages and challenges, and the choice depends on the specific application and target cells, after that making the cut. Once the CRISPR-Cas9 complex is inside the cell, it needs to locate the target gene and make a precise cut in the DNA. Once inside the cell, the guide RNA directs the Cas9 protein to the target gene. The guide RNA ensures that the Cas9 protein cuts at the correct location. Cas9 then makes a precise cut in the DNA at the desired location. This cut is the first step in editing the gene. Following this, cellular repair mechanisms come into play. The cut in the DNA triggers the cell's natural repair mechanisms. These mechanisms can be harnessed to either disrupt the faulty gene or insert a corrected piece of DNA. Scientists can exploit these mechanisms to either disrupt the faulty gene or insert a corrected piece of DNA. This step is crucial for ensuring that the desired genetic changes are made accurately, finally monitoring and verification. After editing, the cells are carefully monitored to ensure that the CRISPR-Cas9 system has worked correctly and that the desired genetic changes have been made. This involves rigorous testing and analysis. This step-by-step process highlights the precision and elegance of CRISPR-Cas9 as a gene editing tool. Each step is meticulously planned and executed to ensure the highest level of accuracy and effectiveness in gene editing. Section 5, CRISPR. Success stories, real people, real results. The promise of CRISPR-Cas9 is becoming increasingly real as we witness its life-changing impact on patients with previously untreatable diseases. One such story is that of Victoria Gray, a young woman who suffered from severe sickle cell disease. Victoria endured excruciating pain crises and frequent hospitalizations throughout her life. In 2019, she volunteered for a clinical trial using CRISPR-Cas9 to edit her blood stem cells. The results were remarkable. After receiving the CRISPR treatment, Victoria's debilitating pain crises completely disappeared. She no longer required blood transfusions and was finally able to live a normal, healthy life. Victoria's story is a testament to the transformative potential of CRISPR-Cas9. Her case, along with many others, provides hope for countless individuals and families affected by genetic diseases. Section 6. Beyond Medicine. CRISPR's potential in agriculture and the environment. While CRISPR's applications in medicine are groundbreaking, its potential extends far beyond treating diseases. In agriculture, CRISPR is being used to develop crops that are more resistant to pests, diseases, and harsh environmental conditions, Imagine crops that require less water, fewer pesticides, and can withstand the challenges of climate change. CRISPR is making these possibilities a reality, paving the way for a more sustainable and food-secure future. In the realm of environmental science, CRISPR offers innovative solutions for tackling some of the world's most pressing problems. Scientists are exploring its use in cleaning up pollution, developing biofuels, and even controlling invasive species. CRISPR's versatility and wide-ranging applications make it one of the most transformative technologies of the 21st century, with the potential to revolutionize multiple sectors for the betterment of humanity. As we marvel at the limitless potential of CRISPR technology, it's crucial to consider the ethical implications that come with such power. Editing the very fabric of life raises profound questions. Who decides what changes are acceptable? What are the long-term effects on biodiversity and ecosystems? And how do we ensure equitable access to this transformative technology? Governments, scientists, and ethicists are working together to create guidelines and regulations to navigate these complex issues. Public dialogue and transparent research are essential to balance innovation with responsibility. The future of CRISPR holds incredible promise, but it also calls for careful stewardship. By addressing these ethical challenges head-on, we can ensure that CRISPR's benefits are realized in a way that respects both our natural world and our shared humanity. Thank you for watching. If you enjoyed this video, please like and subscribe to our channel for more content on groundbreaking scientific advancements like CRISPR-Cas9. Stay tuned for more updates and exciting discoveries.